All right, students, these are the notes on genetics, the introduction to genetics, part one. You'll get a part two of these notes. This one is really taking you back, the history of genetics, really the beginnings of what our modern genetics or the science of genetics is. And that teak down there is the same one I showed you the other day before the class traits lab. That's actual strand of DNA right there, which you're seeing in motion. Um, I wanted to show you this slide because we need to review a little bit about the cell and where the DNA is located, where that genetic material is located. If you guys remember, the nucleus houses that DNA, right? Houses the genetic material, the blueprint for what makes you the individual you are, the species that you are. So if you see that this part right over here, right? The whole cell, the nucleus of the cell, and you see the chromosomes in the nucleus. If you took one of those chromosomes and enlarged it, and you saw one strand like this, and you started to unwind that chromosome, you'd see that same double helix, is what they call it, the spiral step, spiral staircase like structure that I've got a few here on the side for you to see, is the DNA. And if you were to enlarge that even further, you would see that those steps, right, are the actual genes. So the segments of the DNA are the genes. There's some letters over here on the one side, AGTC, and those stand for what are called chemical bases, adenine, guanine, thymine, cytosine. Of course, you don't see the letters in the DNA or on the genes, but it's the way those chemical bases are arranged along that strand of DNA along the genes that really determine which traits you're going to have, which traits you've inherited. It's all in the sequencing of those bases. All right, I wanted you guys to watch this 4.5 minute or so video on Gregor Mendel. He's a pioneer of the science of genetics, and what he discovered in the 1800s was revolutionary at the time and has stayed with the science of genetics ever since then.
Between 1856 and 1863, Gregor Mendel, an Austrian monk, grew and studied peas. That probably doesn't sound that exciting, but what he learned about genetics changed everything. Let's look at what he did. Mendel bred pea plants, looking for many different traits like color, or wrinkly or round seeds, or tall or short plants, among other things. Once he had a purebred trait, he would run an experiment. Mendel crossed two purebred traits. We'll look at green versus yellow-colored peas. When the offspring grew, he found that all of the peas were yellow. He couldn't understand why the green trait disappeared. Why didn't the peas have a 50-50 chance of being green or yellow? To explore this further, he took two peas from the first filial, or F1 generation that was just made, and he bred them together. Yes, this is inbreeding, but they're peas, so no one seems to mind. When the second filial generation arrived, F2, 75% of the peas were yellow, but 25% were green. The green peas seemed to disappear in the F1, but magically reappear in the F2 generation. What could be happening? Mendel decided that there were factors, which we now call genes. The genes could be either dominant or recessive, and dominant alleles or traits mask recessive alleles. In this case, the yellow is dominant, and the green is recessive. So the purebred parents made offspring that had green and yellow alleles, but only the yellow showed because it was dominant. And when the F1 generation bred, there was a chance that they could both give the recessive allele to the offspring, so the green peas showed up again. If you could look at the chromosomes in the pea plants, you would find the gene for seed color. A gene is a section of DNA that controls a trait. The different seed colors have different alleles, which are variations of a gene, like green or yellow. Allele is a fun word to say, and it's actually even more fun to write in cursive, but you can try that next time you're bored. Now you get 2 alleles for every gene, because you have a set of chromosomes from mom and a set of chromosomes from dad, and the same is true for plants. When you write alleles, dominant alleles get capitalized letters, and recessive alleles are lowercase. If a dominant and recessive allele are together, it's called heterozygous. Hetero means different, and the alleles are different, so that makes sense. If the alleles are the same and dominant, we call that homozygous dominant. Homo means same. If the alleles are the same and recessive, we call them homozygous recessive. The letters we choose here are arbitrary, but it's good to use letters that are very different when you view capital and lowercase versions, otherwise it can be confusing. So letters like O aren't good, but letters like Y, T, or R are great. In pea pods, green is dominant over yellow. If these are the genotypes or allele combinations, what are the phenotypes? Having just one dominant allele of the gene will make it green in this case. So there are 2 green pods and 1 yellow pod. Now this is pretty simple, but let's try thinking about this in terms of making gametes. Assuming tallness is dominant, what would the genotype look like for a man who's homozygous for tall? And what are the possible gamete combinations? Now because tall is dominant, this would be a homozygous dominant genotype, 2 capital letters. Again, you can choose any 2 letters you like, but I'm going to use T for tall. When gametes form, only half of the DNA is in each cell, so only 1 allele can go to each of the sperm that are formed. There's only one possible gamete combination in this case, capital T, the tallness gene. But let's try another case. What would the genotype look like for a woman who's heterozygous for tall? And what are the possible gamete combinations? Now, heterozygous means different alleles, so this will be written with a capital and a lowercase t. This also means that there are 2 possible gamete combinations, an egg with a big T for the tallness gene, or an egg with a little t for a short gene. Heterozygous traits will have more variation in their offspring. Thanks for watching this episode of Teacher's Pet. Don't forget to like and subscribe, and follow me on Twitter at SciencePet.
All right, so now you know something about Mendel. He's often called the father of genetics because of those things that he discovered. He identified dominant and recessive traits by studying the heredity of pea plants. And I put in some passages from the book on the next few slides. This one says, what is heredity? Of course, I have the read function on here. So it gives you the definition of heredity. Some things about his experiments and some background, and you just heard some of that in the video, the short video, but it shows some of the traits of the pea plants that he was really focusing on: seed color, seed shape, color of the pods, the color of the flowers, the position of the flowers. He started to see that some traits showed up more than others, and so that he called them dominant, right? And he saw that some kind of faded away and didn't show up sometimes, and so he called those recessive. And he wasn't using the word genes, but he was using the word factors. He knew there was factors involved, and it was going to be 2 factors for each trait. And what he was thinking about there is what we would later know as genes. But he made these crosses, right? And it says parent plants. Mendel crossed true breeding green pod plants with true breeding yellow pod plants. In this case, he was looking for the pod color, right? So true breeding meant, and if you listen to that last page, it goes into what that means. But so that means they're purebred. So the green pea pods had the 2 factors or genes or forms of the gene for green, while the yellow pea pods had the 2 factors or forms of the gene for yellow color. And when he crossbred those two, he actually manipulated the breeding. So what he did was he probably used like a little paintbrush or something and took pollen off of one flower and spread it onto another flower, and that's how he got the first generation. It says all first generation plants had green pods. So he was seeing that even though he had bred the two true breeding plants for the pod color, he was getting only the green color. That's what got him thinking about, this, there's something to this. There's a dominant color. And it wasn't until he let those self-pollinate and ended up having the second generation that he started to see the yellow color again. Still only 1/4 of the time, but he started to see those. And that's when he was saying, okay, that's a recessive trait because it's only showing up some of the time. It's being masked in that first generation, even though these first generation are carrying those factors or genes, they're not showing them as a trait. So dominant was showing over the recessive. They started to show up in this second generation. And then I gave you a few definitions here. Heredity, genetics, traits. Those are going to be important vocabulary words for you guys. And the last one says traits are an organism's physical characteristics. So I wanted to give you some examples of those. All right, so some examples would be something like eye color. And eye color is not determined by just single genes like in the class traits lab that we did. There's multiple genes involved and combinations and blending, and so you see a lot of different colors. Everybody thinks it's just brown, blue, or green, but really there's a variety of colors that come from those. Hair color is another one, kind of works the same way. There's a lot of blending that goes on there, multiple genes. Blood type, right? We don't see that, but it is a physical trait. That's one of those that's actually physical, but not outwardly apparent, but it is something you inherit. And height is another one. And I thought I would give you this poster on here that shows most popular Marvel movie actors and their height comparisons, and got quite a range there of different heights of the different actors, including Stan Lee, the creator of the whole Marvel industry, basically. All right, then you have some slides, some stuff to answer about some of those first couple of slides. Where are those chromosomes located? What are the segments of the DNA that Mendel called factors? So you should be getting those. Another slide with some questions. One more with questions. And then I want you to give me 3 examples of traits with pictures that are not listed in the notes. And they don't have to be human. So type in whatever the trait is that you want to use, and then give me a picture here. Find some on Google or whatever, copy them and paste them in this place, make them small so that they'll fit in that space. That's what I want you to do with those. Could be any traits, could be any organism. Right? It doesn't have to be a human organism. All right, guys.